a new gene therapy for one of the most common forms of congenital blindness was safe and improved patients' vision, according to initial data from a clinical trial led by researchers at the University of Pennsylvania. The therapy delivers working copies of the gene GUCY2D to the eyes of patients who have severe vision impairments caused by mutations in the gene. Each of the first 3 treated patients experienced the improvement in some aspects of vision without serious side effects, according to the new study published in the journal Eye Science. The researchers say that they found sustained improvements in both day and night vision, even with a relatively low dose of the gene therapy. The GUCY2D gene is one of about 25 different human genes whose mutations cause problems in the retina, leading to severe vision impairment from birth or early childhood. This family of inherited retinal disorders, collectively known as Leber congenital amaurosis (LCA), accounts for a considerable portion of blindness in children worldwide. Normal copies of GUCY2D encode an enzyme in the key pathway that light-sensitive rod and cone cells in the retina use to convert light into electrochemical signals. A lack of this enzyme blocks the recovery of this pathway, preventing the reset needed for further signaling. As a result, the signal from rod and cone cells becomes very weak, which equates to severe vision loss. Even in adults who have lived for decades with this condition, it is often the case that many light-sensing retinal cells remain alive and intact despite their dysfunction. Thus, adding functional copies of GUCY2D via a gene therapy could get those cells working again and restore some vision. In the year 2019, the researchers began the first clinical trial of a GUCY2D gene therapy, a solution of a harmless virus that carries the gene and is injected beneath the retina, initially in just one eye per patient. They are following each patient for 2 years after treatment. In the new report, they described their findings after 9 months in the first 3 patients treated. The first patient experienced a substantial increase in light sensitivity in rod cells, which are more light sensitive than cone cells and are chiefly responsible for low light or night vision. This patient also showed improved pupil responses to light. The second patient showed a smaller but sustained increase in light sensitivity in rod cells starting about 2 months after the gene therapy. The third patient showed no improvement in rod cell sensitivity but did show significantly improved visual acuity over the 9-month follow-up period, an improvement that the researchers tried to better function in the patient's cone cells, the predominant cells for daylight and color vision. The researchers say that these initial results from the first ever trial of a GUCY2D gene therapy are very encouraging. There were no serious adverse side effects, and any side effects that occurred in the patient's retinas resolved. The gene therapy dose used in these first 3 patients was the lowest of the doses the researchers plan to use in the study. So they are hoping to see continued safety and greater efficacy in later enrolled patients who will receive higher doses.